fondly remember our fun breakfasts and lunches for research discussions. That was amazing. That was a training I always cherished. His academic brilliance and dedication to the field is an inspiration, and I continued after learning from him in the same area. I will now hand over to Bill, otherwise, I will encroach on his time. I had lots and lots to tell about Bill. So, over to Bill to deliver first plenary lecture of IAN 2020. Over to Bill. William J. Schwartz.
Thank you. Thank you so much, Anita, Neeraj, and the Indian Academy of Neuroscience for inviting me to this great meeting. And good morning, afternoon, evening, good night to everyone. And I appreciate those incredibly kind words. And so let me share my screen. And let's see, I guess it helps the internet to stop the video. So thank you very much. I'm honored to give this plenary talk. I realize that I've reached an age where now I can give some advice, even if no one will listen. At some point, it's okay to give advice. So I thought, if you'll permit, that I'd give a bit different talk for our first plenary, and that is some unsolicited advice about publishing and about impact. So we're all familiar with how we've been measuring impact these days. I'm going to use, no, not this, little pointer. So it's become routine that impact is equated with so-called high impact journals. You see the proliferating ones here, sometimes called vanity journals by Jeff Hall, previously of Brandeis, luxury journals, Randy Sheckman from the National Academy of Sciences. And this impact factor mania, I'm going to call, is related to the IF, the impact factor, which, as you know, is a bibliometric tool that is being used to rate both researchers and the quality of their individual research papers. And the problem with this, first of all, some of you may not know that that was not the purpose for which the impact factor was invented by Eugene Garfield. It was originally conceived as a tool for librarians so that they could make decisions, financial decisions, on which journals to purchase. That was the conception of the impact factor. And we could argue that in the time since then, 1972, the impact factor has worked to, I'm going to say it, distort the scientific enterprise. It, of course, is based on the journal where the paper is published, not by the content of the paper itself. It really is a ranking of journals, not scientists or papers. And you'll also realize that the impact factor represents the calculation of the number of total citations in a per year, divided by the total number of citable articles. But the impact factor is only for the previous 2 years. So it's a rolling average of 2 years. So truly innovative research, which takes some time to appreciate, takes some time to confirm, doesn't usually contribute to the calculation. So the question is, why are we still bothering with this? Maybe it gives scientists more exposure to publish in these journals. Actually, it turns out that the higher the impact factor, the longer is the interval from initial submission to publication. So scientific communication is delayed. So if there is exposure, it certainly is not coming early when people can make use of it. Here's some data on that. Here are the PubMed papers, which are published in 2013, and suggested journals with the lowest, the actual lowest impact factors, but also the highest impact factors here have the longest review times. So here, for a journal with an impact factor, say, of 5, it's about 3 months from submission to acceptance, and that increases to something in the range of 5 months for these. There are a number of reasons for this. One is a reason that I think us oldsters will recognize, and that is the amount of information, the amount of data which is required for submission and acceptance in these journals has increased over the years. Here's an interesting panel. This is the number of panels in figures. You know, the number of panels that compose each experimental figure, compared 1984 to 2014 for Cell and Nature. Here's 84. The number of panels have skyrocketed in both of these, not only in the paper, but also in the supplemental data, which had hardly existed before. So that's a hint that the amount of data required per paper in these journals has increased. Well, maybe you'll get more citations. Maybe that's the idea. But you know, the citation frequency for the journal doesn't predict the citation frequency. I'm getting that. Doesn't predict the citation frequency for individual papers. So the IF is characteristically determined by a minority of highly cited papers. Here's what I found was a really interesting table from LaRiviere. So here they took a smattering of journals, multidisciplinary often journals. Here's the impact factor. So it ranges from impact factor of 38 or 34 for Nature and Science to 3 for PLOS One, a little less than 5 for the Proceedings of the Royal Society, 2 for General Informatics, and everything in between. And this is the number of citable items, that is the number of papers which actually have citations below this impact factor. For all of these, the majority of papers are well below this. It doesn't matter, really, what the journal impact factor is. Something like 65 to 75% of papers are cited less. Here's Nature. So there are some papers that are cited highly, but like all the journals, most of them are cited modestly. So for all journals, there are a large number of papers with few citations, relatively few with many citations. So I would say neither exposure nor citations are sort of very convincing reasons for these. So what should we do? So where's the advice? Because all of us will still be sending our papers there. And so I thought I might give 2 stories from the history books. I call it Tales of 2 textbook chronobiologists, circadian biologists. Certainly the circadian folk are aware of these 2. Jean-Jacques Dortu de Marin and Kurt Richter. So from the 17th, 18th centuries to the 20th century, I thought I'd give two stories that would illustrate perhaps some ideas about how to make an impact. So I only have two pieces of advice. The first piece of advice is to share with smart friends, which is something that we often are loath to do because we're holding on to our data so that we can send it to a high impact journal. And I'd like to tell the story of de Marin. He was actually in his day a world famous scientist who very few, if any, would remember. Actually, the circadian biologists may remember him the best, even though that wasn't his field. He was an astronomer, a mathematician, geophysicist. He was elected to the French Royal Academy of Sciences, one of the two oldest scientific academies in the world. He was its permanent secretary, its leader. He was a fellow or member of all the learned societies in Europe. He was the editor of an early journal, along with Philosophical Transactions. This is sort of one of the two earliest scientific journals. And he was a frequent visitor with the glitterati, the intellectual group in Paris, in Madame Tancin's salon, where, along with, for example, Voltaire, he was one of her sages or her experts. He grew up in the south of France. There, he had a vineyard. The vineyard still exists. Here it is today. They make wine, the Domaine de Marin. They make all sorts of wines, but they're famous for their rosé, which they call the Aurora Borealis, which was a favorite of his. There's a crater on the moon named after him. So here's a guy you've never heard of who was a leading scientist in the 18th century. The French Academy was founded in 1667. Louis XIV was the King of France. And here's a painting showing the original members, and Colbert, here's the king. And it was organized into 6 sections. They were all independent sections: math, mechanics, astronomy, chemistry, botany, and anatomy. You can see there's no medicine. Medicine was really botany, right? Because botany was concerned with not only the cataloging of plants, but also the possible discovery of therapeutics, pharmacotherapeutic substances. The Academy put forward a journal once a year. This is the year 1729. And it was divided into 2 parts. One was called the Memoir, and these were regular papers that we would recognize by the authors. And in this issue from 1729 is a paper from Monsieur de Marin on geophysics and astronomy, new conjectures on the cause of the diurnal movement of the Earth from the east east to west. And it says by de Marin, it gives a date. One of the things about these papers that is notable is, here's a piece of it, is that it was written in the first tense, the personal tense. My conjecture, I think, my explanation, and so on through the paper. There was a second part of the each issue, which were the histoires. These turn out to be, I learned, more like modern news and views, or kind of a newspaper. So these were short reports very short, of like a page or two, that were based on the sections of the academy: physics, anatomy, chemistry. Here's botany, and here's a little entry, Observation botanique, a botanical observation. This is the paper, not too long, which arguably set off a field, which 3 years ago was honored with a Nobel Prize. This paper is about a plant. The one in particular was mimosa, which, like other plants, during the day with lights on, had its leaves open, and at night they were closed. And a little translation of part of this is here. I translated it. But Monsieur de Marin observed that for this phenomenon, that is mimosa's day-night difference, for this phenomenon to take place, it's not necessary for the plant to be in the sun or outdoors. When it's always kept in a dark place, it still opens up quite perceptibly during the day and folds away or closes for the night at the same time every evening. The experiment was performed towards the end of summer, repeated several times, and here it is. Here's the sentence: the sensitive plant senses the sun without seeing it in any way. He didn't say there was a clock. He just said that the plant was aware of the sun without it being exposed to it. Then comes a curious sentence. It's reminiscent of that delicate perception by which a large number of patients in their beds can tell the difference between day and night. And ends near the end with the notice that the ordinary occupations of Marin prevented his pursuing his experiments, and he's happy, he contented himself with a simple invitation to botanists and physicists who themselves might have other matters to pursue. So there are some curious features of this. Obviously, one is it's written in the third person. It's describing de Marin. He didn't describe what he was doing. It has a little sentence about a medical implication that's hard to imagine why Marin would know about that. And then it's not Marin that's making the invitation, but something or someone else says that he is extending it. So, as others have commented, it does not appear that Marin wrote this paper. He gets credit for the experiment, but he didn't write it. He wasn't the one who popularized it. Who was it? We don't know for sure, but the clue is on the next page is a report of a plant by a Monsieur Marchand. Who was Jean Marchand? His father was Nicolas Marchand, who held a degree in medicine from the University of Padua, which, as many of you know, was the leading medical school possibly in the world, certainly in the West. He then became an apothecary, basically a pharmacist, to the Duke of Orleans. The Duke had a botanical garden at his chateau that was managed by the Duke's physician. So here are two docs, Marchand and the Duke's physician, who are hanging out together in the garden, talking about patients, presumably, and plants. Nicolas Marchand was a founding member of the Academy. He was the botanist. He was named director of the King's Garden, and he was instructed to compile an encyclopedia of plants. His son was Jean Marchand. He was elected to the Academy upon his father's death and director of the garden. species from all of Europe were sent to him in 1 year alone, 500 species. And then, in a move that some of us will recognize, the Academy actually terminated the Encyclopedia Project, and Marchand lost his pension, his support, and his position. But he continued, as a historian remarked in the beginning of the 20th century, to prepare botanical descriptions of great exactitude. And look at this, even wrote the following: in the month of July 1715, Monsieur Marchand noted in his garden a plant which he did not know, but the principal reflection he had upon these two plants is that it would not be impossible for new species to be formed. This was in 1721. This guy was something. He also served on the review committee that selected these short pieces in the Academy's journal. I've been unable to have a look at him, but I'm imagining, as others have, that Marin and Marchand, recognizing each other's genius, are sitting in the gardens, talking about their interests. And of course, Marin is interested in the sun and the stars and the earth, and he's curious. And for whatever reason, takes his little potted plant indoors and describes this curiosity to Marchand, who recognizes the importance of this and chooses to write about it. And today, Marin is known really for this, not for his astronomy or physics. And so his impact was cemented, I propose, by sharing with a smart friend. The second piece of advice is to publish. And I say this because in this day where there's abundant social media, where there's abundant sort of merchandising of our science, where as we wait for our papers to get into high impact journals, sometimes over years, sometimes the projects are dropped, sometimes projects are not published if we don't think they could get into a high impact journal, or it takes so long that it may be lost. And so some simple advice is to publish. And here's my second story about Kurt Richter, who was a famous American psychobiologist. And I see there's a chat, which I guess, should I open that and just check it out to make sure nobody is mad at me for anything? Thank you very much. Okay, let me return to my pointer. So Kurt Richter was famous as an American psychobiologist, interested in what we would now call neuroscience. As was pointed out in our welcome, the field of neuroscience is a relatively new moniker. And he was dedicated to the study of innate or vegetative behaviors, if you like, that serve homeostatic regulatory functions. Here he is in 1920. And in 1919, he arrived at Johns Hopkins as a grad student of John Watson, J.B. Watson, right? The behaviorist, the famous behaviorist, J.B. Watson. 1 year later, Watson was fired for some, how can I say it? Some indelicate relationship with a graduate student who he later married. The year after that, Richter was given a PhD. The year after that, he was head of the laboratory. Not bad. So in 3 years, he went to a first-year graduate student to head of the laboratory. And in circadian biology, Richter is sort of notable for popularizing two tools that have had widespread use. One is what we would now call the running wheel, which he called a revolving drum here. And here you see he shows an activity rhythm of a rat. On the top is the light-dark cycle drawn out this way on the x-axis, alternating light and dark, night and day, and then continuous darkness. And here is the activity in the revolving wheel. And this continues with a circa daily rhythm, a circadian rhythm in darkness. And when one looks at this 55-page monograph to find out what these wheels are all about, this is what I found. Ordinary revolving drums were used, and then a very detailed description, 12 inches diameter, 10 inches wide, revolved very easily. Maybe they would have 12,000 revolutions in 12 hours. But what is an ordinary? What's ordinary about this? I mean, does everybody have a revolving drum around? Well, actually, the original revolving drum was a PhD thesis by a fellow named, who no one has heard of, probably in this Zoom talk, Colin Stewart, at the time at University of Toronto, where he described this. Here are the pens. You know, he was using it for other kinds of physiology and described the construction of this drum. Later on, 4 years later, Richter published a specific paper about his apparatus. Here's what we probably now call running wheels. And he there cited Stewart. 4 years later, cited Stewart. Who was this guy? So he got his bachelor's in 1894, University of Toronto, a PhD at Clark University, which is in Worcester, Massachusetts, close to where Professor Anita and I worked together at the University of Massachusetts. And then it doesn't look so good for Stewart. He has odd jobs, which he doesn't seem to be able to hold. A year at the Harvard Medical School, 2 years at Columbia in New York, a year as this is a wonderful title, a sometime fellow in biology. I love this. Maybe I could be a sometime professor. And then 3 years in University of Pennsylvania. This does not look good for Stewart, but fate smiled upon him, and he was awarded an associate professorship at Dartmouth Medical School in New Hampshire, and was there until he died in 1944 as a named professor of physiology. He's written up in Tenney's Brief History of Physiology at Dartmouth. For 40 years, Stewart taught a 2-year physiology course. First year was lecture, and the second year was the lab. He was described as gentle and serene, barely audible. Interesting. Doesn't make a nuisance of himself, so that Richter can take his drums. Later, in fact, he developed Parkinson's disease. He was one of these guys who was ambidextrous, and would do anatomical and physiological diagrams using both hands, and was always dressed for the part. Now, the other innovation, as we say, or popular innovation of Richter, was the actogram. And unlike what he showed in 1922, unlike this, which is how we'd ordinarily draw a time series with time on the x-axis, he put it vertical. And so now days, succeeding days, are aligned from top to bottom vertically. And this is called an activitygram, or actogram, has been very useful for the visualization of circadian rhythms, as you can see, because it's easy to see changes in phase or period. Well, actually, the first actogram was in a PhD thesis. And this is by a fellow named Maynard Johnson, who I suspect no one here. I saw Sato Anma here, she knows, but perhaps no one else. He studied deer mice, and he recorded their cage movement. So the cages were suspended, and when the animals moved around, the cages moved, and he had paper on metal discs, 2 discs. It rotated over 12 hours. It was either a pen on paper, or he smoked the paper with a pinpoint, and then he put it in shellac to preserve it. And then in the methods, he said he did this. He cut them out and scored them like this. He didn't say why he did this or how he came up or what the justification was, but he did it. And he constructed in 1926 actograms where he did the kinds of experiments that people do today. Here's a mouse in a light-dark cycle outside, inactive, active. Here's constant dark in the lab. Here he's put them in the lab, put light at night, and then put them in the dark, phase shift by light, persistence of rhythm, free running rhythmicity, the whole thing. Who was this guy? So he graduated from Bates College, one of the premier New England small liberal arts colleges, where he was in the Honor Society, Phi Beta Kappa. In the yearbook, he's listed as a progeny of learning, and what was written about him by other students. Maynard is not avaricious, but he does love lady scientists' fudge. He is ready to try anything or anyone once, from a moonlight waltz to a sermon. Now, I've tried, without avail, to see if in the 20s in the United States, a lady's fudge did not refer to candy, but I haven't found anything. But we see beyond that it looks like he's brave and creative and kind of courageous. But, maybe the once is important here too. He'll try anything once, but will he stick to it? He got his PhD 4 years later at the University of Illinois in zoology. And then at some point was at the biological laboratories at Harvard University. And in a 1939 paper, he thanks 2 individuals that they lent him their laboratory space and equipment. So it's really unclear what he was doing there. I've called the biological laboratories. I've looked, I've had people look. They don't have any record of him. But that's where he published a couple of later papers. And then suddenly he left. He became a wildlife technician at the Fish and Wildlife Service, now the National Park Service. Rose through the ranks, assistant refuge man and the refuge manager at a refuge in North Carolina. He's lost for a little bit, and then I found him again as the U.S. Army civilian entomologist in Japan. He was then an entomologist in the U.S., He retired from federal service in 1968. So these two are here in Hyderabad, not forgotten, and a tribute to two published PhD theses. Here was an impact, the running wheel and the actogram, from two people who did not publish in high impact journals and whose names we don't readily associate with neuroscience or chronobiology. But here they are. Here's Stuart, very natty, quiet, unassuming. And here's Johnson in his yearbook. Yeah, he does look like somebody who would try anything once. Maybe he irritated people, I don't know. And later on. So what was this all about? Well, you know what it's about. It's about standing on the shoulders of giants. And all of you know Sir Isaac, who wrote in a letter to Robert Hooke, a sort of competitor of his, "If I have seen further, it is by standing on the shoulders of giants." Here it is, actually. Here's the letter. Here, it's blown up. Here it is. "If I have seen further, it is by standing on the shoulders of giants." And then he goes on. And then he sort of makes somewhat snide comments. Yeah, actually, that had already been published by John of Salisbury in the 12th century. We are like dwarfs sitting on the shoulders of giants. We see more and things that are more distant than they did, not because our sight is superior or because we are taller than they, but because they raise us up and by their great stature add to ours. Wow. Who knew John of Salisbury? So I've reached my 45 minutes, as Professor Anita advised. And do I have, can I codify any unsolicited advice for the youngsters and the oldsters on making, and everyone in between, on making an impact? And I would say, please be generous with your shoulders. Aryans Kappers, a Dutch neuroscientist who really did critical work on the pineal gland, wrote, "The stream of life continues while the life of individual man is restricted. For a great deal of the work done by scientists is based on that of their predecessors. In the best case, they may contribute a little to a better knowledge of facts which can help man to understand better the world he lives in, and which may be of some practical use for his fellow men. Considering this should give a sense of modesty and humility, but also a sense of great happiness." And with that, I will unshare myself and appear. Now the sun is in my face. And so I want to thank the Academy, Adita, Neeraj, and all for, and Shashibala, and all for organizing this meeting under unusual circumstances and pulling it off. Thanks very much.
Well, thank you very much for this wonderful lecture and the journey you took us through from very, very old stories about how simple ideas in a PhD thesis become used for centuries to come. Thank you very much for that fantastic lecture. I appreciate it very much with this little bit of a clapping as we would
Thank
do.
you.
And I just wanted to say that, yes, it's absolutely true. Before we take on, I guess we do take on questions from the audience, and I hand over to Professor Jagota, is when I talk to my younger students, one is a comment. As soon as they join for PhD or something like that, they just do not recognize how incremental is the progress in science. That's what you showed in your last slides. And it takes them a while to get over that frustration. So that was fantastic. The second thing, a comment that I hear from my students, when I tell them that, you know, it's important to publish good science rather than in the biggest journal, they say, you know, you're all gray now, earned or otherwise, and you can say whatever you want to say. We are young, we have to set up our career, and unless we publish in big journals, nobody is going to notice us. Would you like to comment on that?
Yes, and of course there's some truth to that, and all of us strive for that as well. We're all under this impact factor mania. I guess my two comments would be, okay, it's okay to try, but I know laboratories that have sort of held on to their data, which could be citable, which could be useful, for years, where it might be helpful for others. Now, of course, with bioRxiv, there are ways to sort of get it out. But nevertheless, I think it is important sooner rather than later. I mean, one puts a time limit on it because nowadays I would say to them, if you're interested in the exposure, there's plenty of social media. You can tweet all you want about your bioRxiv paper if it needs to go out. But it's still important, I think, still to publish.
Absolutely. I remember my postdoc mentor, Professor John Kass of Vanderbilt University. He always used to tell me, at some point, you have to say that's it and send it out. There is no end to it otherwise. I will hand over to Professor Jagota for her comments and maybe take on the questions. Professor Jagota.
Yes. Thank you, Professor Neeraj, sir. And thank you, Professor William J. Schwartz, for such a wonderful lecture. And I really thoroughly enjoyed. And of course, I believe everybody else has enjoyed. And the questions and the story which you took through, all of us are toughly facing in today's generation more, because when we were doing postdoc, and I still remember Bill will say, let it curry, means like let means let it take shape, and then we will publish. But as Professor Neeraj pointed out, these days this is the toughest thing, at least I do not know across the world, but in India, in our laboratories, we face that students just join, and next 1-2 months they think like something should go, and we do not, they just want some publication so that they jump to the next platform, and once they jump to the next platform, they don't want to finish the previous task even of finishing because they are now engaged in the next. So how to solve this issue? Can you please advise?
Well, as we all know here, sort of the mentoring and advising of our students is really sort of a critical part of this. And, I do say, and we all know, that our reputation, and as Neeraj pointed out very clearly, our reputations as scientists slowly build over decades. And presumably, we will have more ideas than just the one we're working on now. Some will pan out, others won't. And one of the most difficult things I find for all of us is that issue, is what John Cass said, is when do you stop, move on? When is that mountain ahead of you not the promised land, but just the Himalayas beyond? So, how do you make that decision? And that's the beauty of, stories and experience that we can give to our students. And, maybe a small mix of short observations that can be published, presented in a forum, because still, and it'll come again, we all meet each other at meetings, we have our students present, and they make an impact there on others, where they hear, where they can meet others, people can see. Picasso didn't become Picasso until decades after he had mastered all of the techniques of art. And we're artists in that way too. So I think we do actually have to state what's obvious to us, that it really takes decades of reliable contributions to make ones known. But I agree with you. In this world, it's a tough assignment.
Well, I think you have given us a great message. Professor Patro wants to ask. Please, sir, go ahead.
Well, don't you agree with me if I say that instead of asking number of papers or the impact factor number, we start asking the content of the papers? Then the students would not do this. They'll start looking into the content of the paper more than the number of publications or the impact of the publications. The only thing that we need to do as leaders of science is we stop asking how many papers you have published instead of asking what you have published. Instead of asking what impact factor you have published, we should ask how many people have cited you, how many people have appreciated your work, or how many people have really liked reading your paper. That should be the question, instead of asking the number of papers or the impact factor number at which you are published. That's my feeling. I believe that should be the way.
Well said. Well said. I might say, it reminds me that in terms of the impact factor, as far as I can make out, the only ones who like the impact factor are administrators. Hopefully there are no administrators here. The journals don't like them. So I had lunch with the editor of Nature something or other, Nature Neuroscience or one of those, and she said they hated it because every year they were in this arms race with others, and it was driving them crazy. So somehow, and there are papers. There are many papers. This is known about the impact factor and how it's being misused, and yet, because it's easy to put it in a spreadsheet.
Thank you.
I would like to add to Professor Ishan Patro sir's comment that even people are not like, if it is a paper which is published few years ago and has built up citation index, after many people have worked on it, is considered different impact than these days by using paid journals and open access, where you have already paid and open it to the world. That particular paper may get cited fast as compared to the traditional. So it is very difficult which metrics should be used. And I once again agree with both Bill Schwartz as well as with Professor Patro, the content of the paper, the techniques, the ideas, I think while judging a candidate, that should be the metrics, because others are numbers. In even like impact factor is a different kind of a number, and citation index built over years means people are continuously working on those ideas. Some publications these days, which are especially in the like open access journals or these, they get citation also very fast, not because because that is just immediate hot topic. We do not know that like immediate movie number. So so and after like a little while that idea subsides, but that particular year you have citation, you have this. So an idea which has built up over the years and people have worked on it, carried it further. I think, Bill, that what is your comment on that? That should be looked into.
Well, yeah, I mean, I don't know about in India. I suspect the same in the US. Expertise, individual expertise is considered subjective. And people can do very fancy statistics with quantitative data, even though the data itself may be not so useful. So this I see everywhere. It's a reaction to a time when certainly there were a cabal of sort of a few people who are dictating the truth. And so now it's swung the other way. And those kinds of things are discounted or considered subjective in the favor of these numbers, even though, as we all agree, the numbers have critical faults with them. I see a hand raised from Professor Seth.
Yes.
Yes.
Over to Professor Seth.
Can I just make a very small comment?
Sure.
And what I feel that why this impact factor got so much importance, because the evaluation process of the candidates, I can speak only for India, not for US, that is sometimes in the interviews, the number of candidates is high and people interview within a small given time frame. So, and then there's expertise. If you really do not have the expertise among the experts to evaluate a candidate, then they go for these things like impact factor or the citations. But something good work which has been done, I think the importance is this: the expert should be fully aware of that work, who can actually evaluate the work. So the evaluation process, I think, must change. Otherwise, in a short given period, 10 candidates, and if you have to find out which is the best one, then people rely on this, something which everybody agrees, okay, high impact factor, so he's a good one. It's a good paper. That's my feeling. So I would like to have your comment, Dr. Schwartz.
Yeah, no, I agree. I think from an administrative point of view, it's easy. So that's led to its rise. In the US, I would say now I've noticed what's happened, that as candidates are being evaluated, the evaluators are actually making phone calls, which they hadn't done before. And in some places, I was called by someone, they were interested in talking about a candidate, and I said, well, I didn't get your invitation to write a letter. He says, no, we don't even bother with that anymore. We just call and get this. So I think it's reverting in a way to a more artisanal, if you will, customized process. But of course, as you say, this takes time, it takes energy, it takes expertise. it could be waylaid by powerful people would say, who have, an undue influence. So somehow there's got to be a balance between the two. But I will say that publishing or publishers, scientific publishers, and all the things we know about, open access journals, other kinds of, predatory journals, libraries that can't afford it anymore. Publishers say, scientific publishers say, they are facing the biggest change and uncertainty, and they don't really know what to do, frankly, since Gutenberg and the print. And so they're really they're not happy. So at the moment, everyone is all of us are in this kind of dystopian state about scientific publishing. So that's why I agree with everyone who's commented that it really is our job as mentors to teach our students about asking questions, curiosity, making an impact by contributing bricks to sort of this edifice of science, where it really is a wonderful, it actually is a wonderful situation that we're in, that we can do that people can stand on our shoulders.
Thank you. I really, Bill, enjoyed your talk very much, and particularly the two examples which you took, the two stories. Very rarely you find people now telling such things in a simple manner, but yet explaining the point. Thank you very much, Bill.
I'm just going to say that among all the advice that you have suggested for young people, another thing that I'm seeing more and more is they don't read the paper anymore. And they just look at abstracts and few little things here and there, and that patience to read the entire manuscript, entire published paper, that's just gone completely. And they miss out the most important indicators of what's good or bad about the paper in that sense. So this is another change that is happening, as you said, since Gutenberg. That's another change that's happening, is where everything is getting abbreviated into few, tens of characters, or sometimes just a little more. So yeah, I mean, you're absolutely right. It's a big change, and we don't know how to deal with it.
If I may, I was just going to say, just before Anita cuts off the discussion, I'd like to bring one sort of happy story, a very short story, so that we're left with positive about the future. But if someone else, I had a graduate student who was very courageous and brave with trying out new experiments, but as you would say, not so meticulous in her notebook, not so meticulous in sort of how to present the data graphically. And I would sort of be on her case, and she was kind of frustrated and annoyed and come on, granddad, why are you bothering me about all this sort of stuff? Let's get on with it. And since then, she's made a nice career in industry. And after her first year in industry, she called me, actually it was through her husband, and they said, I want to thank you for showing me the importance of all these points, because I remembered them, and it's really made a difference in my job. So we can still make a difference, even if everyone is rolling their eyes at us. I think we can still make a difference.
Absolutely.
So this has been a topic which is close to heart for every scientist and every supervisor. So we had elaborate discussion, and I know still people may have questions, and but in the interest of the time, I have to move on. And I thank once again Professor William J. Schwartz for giving such an excellent talk and interacting through question answers and giving us his advices. And I also thank Professor Neeraj for chairing the session, and all senior scientists, Professor P.K. Seth and Professor Patro and others, for asking such questions which led to such a wonderful interaction. I think Bill wants to say something.
I just, thank you for myself. And also, on my first slide, I guess it's recorded, but on my first slide is my email. But Professor Anita knows it. So I'm happy to carry on our discussion with emails. You could certainly ask Anita for my email.
Okay.
Thank you.
Thank you, Professor Schwartz. Thank you, Dr. Anita.
Thank you.
Thanks again.
Thank you. Thank you. And we give a big hand of applause. And then we move on, as it's on the same link, we move on to the next exciting event, which is K.T. Shetty Memorial Oration. And I invite Professor Harish C. Pant and Professor Bindu Kuti from NIMHANS Bangalore to chair the sessions. Professor Bindu Kuti and Professor H.C. Pant.
Yeah, thank you, Dr. Anita. I'm here. Dr. Harish Pant.
I think we should start because we are so
I'll just give brief introduction to chairperson, Professor Bindu Kuti ma'am, and after Pant sir joins, then you can take over. So currently Professor Bindu Kuti ma'am is working as Professor of Neurophysiology at NIMHANS, and her research interests are meditation and sleep, anxiety disorder, Ayurvedic intervention in sleep, schizophrenia, and sleep. I'm not going to the big list of awards Madam has. I have interacted with Madam at many platforms, and in interest of time, I'm trying to be short. And this I'm doing on behalf of H.C. Pant because sir has not yet joined. She is also in charge professor and coordinator of NIMHANS Center for Conscious Studies. Over to Bindu ma'am.
Thank you, Dr. Anita. Very good evening. And congratulations to both of you, Dr. Anita and Dr. Shashi, for organizing this international online conference. And also thank you from my side for giving this rare opportunity to chair the Professor K.T. Shetty Oration. And I look forward to see Dr. Harish Pant. Now, what should I do? I should, shall I just give some introduction? So, Professor K.T. Shetty Oration, we have initiated in the year 2009 after his untimely death in 2007. And Professor K.T. Shetty doesn't require much introduction because he is a well-known figure among the IIN members, and he was a well-accomplished international neuroscientist and a great teacher, a great mentor, and a very fine human being. I have had the opportunity to work with him, and also I was a good friend of him. While he was serving NIMHANS as a faculty of NIMHANS, as well as a professor and head of the Department of Neurochemistry, and also he has served as the Dean NIMHANS. So it is so great that IIN has actually initiated this oration in the memory of Dr. Shetty as a mark of respect towards his contributions in science, as well as contributions in the growth of many scientific organizations, including IAN. So this year's 2019 year, the K.T. Shetty oration goes to Dr. Anirban Basu, a senior professor from NBRC. And Dr. Basu did his PhD in Institute of Chemical Biology and joined NBRC in 2004. And he has been working in the area of neuroinfection, especially on JE virus. And especially to understand how neurotropic virus cause abnormality in brain function and development, to understand the pathophysiology of infection and inflammation in the central nervous system. Dr. Basu's work not only provides a new and a general principle for cure of the whole spectrum of neuroinflammatory diseases that include infection of the CNS, but also other neurodegenerative disorders that affects millions of people worldwide. And he has immensely published, and with an H-index more than 20. He has been bestowed with many prestigious awards and honors. I will just read out one or two. He has been awarded with the National Bioscience Award for Career Development by DBT in the year 2010. Then elected to Fellowship in National Academy of Sciences, NACI, India in 2011. Then 2018, elected to Fellowship in the Indian Academy of Sciences and elected to Fellowship in the American Academy of Microbiology in 2019. It's my pleasure to invite Dr. Anirban Basu to deliver this oration. And meanwhile, I hope that Dr. Harish Pant will join us. I'm also very happy that I am chairing along with Dr. Harish Pant. He's such a senior scientist in NIH, and he has been a scientist since 1974. And after so many years of his human service, he is retired from NIH in 2019, and currently he is a scientist emeritus in the National Cytoskeletal Protein Regulation Section, Laboratory of Neurochemistry at the National Institute of Neurological Disorders and Stroke, NIMS, NIH, USA. And his major areas of contributions are in the area of cytoskeletal proteins, especially the post-translational modification of these proteins in health as well as in neurodegeneration. With this introduction, I think, shall I invite Dr. Anirban Basu to deliver the oration?
Please invite. Yeah. Dr. Anirban, you can please share your slides.
Yeah.
Yeah. So I want to thank Indian Academy of Neuroscience for considering me for this award. I know it's a very prestigious award, and when I see the previous recipients, I am feeling very privileged. Also, I wanted to wish all of you good morning, good afternoon, good night, wherever you are across the world. Could you please allow me to share my slides? Dr. Anita?
Yes, sir, you already have the permission.
Okay.
हमको कल और परसों
Yes. My slides are visible now? Hello?
Not now, Anirban. I think there's
Not right now.
Not yet.
not yet.
Now?
Yeah, I think they're coming now.
Yeah.
Yes. Just
Okay.
put it on the presentation board. Thank you. Yes.
Okay.
Go
So,
ahead.
as you already heard about Dr. Shetty, I do not want it to go details. He was former professor and head of the Department of Neurochemistry, National Institute of Mental Health and Neuroscience, Bangalore. And he is internationally recognized for his work. And if you see his work in that point of time, it was very novel and fundamental neurochemistry work. He's a great painter, social activist, and fine human being. And for the slides I show about Dr. Shetty, all these slides are kindly provided my very good friend, Dr. Shankar Narayan Rao from NIMHANS. This is one of the Dr. Shetty's photo as a young researcher. And this is, of course, with Dr. Harish Pant's lab at NIH US. I hope he has already joined. So now I move to my oration lecture. So what I will do that my lab is primarily interested in certain aspect of neuroimmunology or neuroinflammation. We use the virus as a model system. So what my title of the talk is Innate Immunity in the Central Nervous System, redefining the relationship between immune system and nervous system. So if you see that until recently, the CNS and the peripheral immune systems were thought to operate independently. You cannot find the term immune system in the index of the current major text in neuroscience, nor the term CNS and brain in the index of major text in immunology. However, new research has led to important advances in our understanding of how immune-related events can influence CNS process, thereby altering cognition, mood, and behavior. It's interesting that last 10 years, there's an emerging literature which showed that even an immune cell like T regulatory cell or Treg can control the cognition and the behavior. Some of the novel work or most seminal work from, say, group like Michael Schwartz from Israel shows that if mice has depleted in T regulatory cell, they have a problem in their cognition and the mood and all other behaviors. The outline of the talk is that historical perspective of brain's innate immune response. Why I decided to talk about this brain innate immune response, there's a reason for that. How the field evolves and recent discovery. And at the end, I will show some data that how brain's innate immune response is seen by neotropic viruses. So this is the historical perspective of glial cell. So around 1856, a German physician or neuroanatomist, Rudolf Virchow, first mentioned about a cell called neuroglia. At that point, he was not sure that cell is a neuronal cell or glial cell. So he had a mixed drawing of the cells, which is called neuroglia. And then this field evolved like Camillo Golgi, Ramon y Cajal, and finally, 1919, 1920, Del Rio Ortega said that there is a cell called microglia. So Ramon y Cajal has this idea that besides astrocytes and neuron, there is a third cell types. He was not absolutely sure. But Rio del Ortega first coined this term microglia and identify a distinct cell type. And Cajal get the credit for the neuron, identifying neuron. Del Rio Ortega get the credit for the microglial cell, and Penfield got the credit for the identifying the oligodendrocytes. That in one slide, I try to put how this glial cell evolved. In these glial cells, out of them, microglia, of course, the most immunocompetent cells in the central nervous system. There are two distinct groups. One is a German group and the Spanish school. So German group belongs to this Rudolf Virchow and this Alzheimer, and finally the Spanish school belongs to the Ramon y Cajal and Rio del Ortega. And Ramon y Cajal is the first person of Spanish origin who got the Nobel Prize in Science. So this is the reason I thought to talk about this topic, because this is the 100 years we are celebrating discovery of the cell as a microglia. And what is the importance of this cell? If you see, it's a fascinating cells. These cells is never been originated in the central nervous system like astrocytes or oligodendrocytes. These cells actually developed in your peripheral system and moved to the brain in some point of your development. But it has an immense role in regulating the normal function of the central nervous system. And this is the year, we are 100 years, we are celebrating this discovery of microglial cells. Ramon y Cajal, 1919, 1920, he has first identified these cells. So this is one of the reasons I thought to talk about this particular microglial cells. The word glia was coined in the mid-19th century and defined as the nerve glue. For decades, it was assumed to be a uniform matrix until cell theorists raised the Newton doctrine, which stipulated the nervous tissue was composed of individual cells. The term astrocytes was introduced in the late 19th century as a synonym for glial cell, but it was Ramon y Cajal, who defined the third element, distinct from glial cells, astrocytes and neurons. He has the idea of the third element, but he was not sure. 1919, Del Rio Ortega introduced the term we use today and thoroughly described both oligodendrocytes and microglia to clearly distinguish them from astrocytes. In a series of 4 papers published that year in Spanish, Del Rio Ortega described distribution and morphological phenotypes for microglia, though Penfield ultimately get the credit for the oligodendrocytes, and Del Rio Ortega get the credit to identifying the microglia. He also noted, it's a very important point, why he also noted that resting microglia transformed into an amyloid phenotype in the defined types of brain disease and pathologies. That point in 1990, 100 years back, actually he had identified the amoeboid microglia, resting microglia, and activated microglia. And if someone is interested, all this Spanish paper actually now translated in English, and GLIA 2016 published all these 4 papers, which originally written by Del Rio Ortega in Spanish. So this is the Rio del Ortega's drawing, and you have to appreciate the fact there was no camera in the microscope that point. Whatever he has seen in the microscope, he has drawn in all the cells. So he has a paper, pencil with him and microscope. So basically whatever he's seen, he have to draw it. And this is his resting microglia. This is the activated microglia. And this is the amoeboid microglia. So that point itself, he has clearly identified 3 different cell types, looking to that microscope at whatever sophistication that point he has, and drawn them. Now, how true this is science-wise? This is the modern day photo image which we have taken in a very highly sophisticated Nikon or Carl Zeiss microscope. If you see here, following a infection in the brain, we have seen the resting microglia, activated microglia, amoeboid microglia. There is a unbelievable, if you see the similarity, what Rio del Ortega drawn 100 years back. That means even without that sophistication, he has clearly identified all different 3 cell types of microglia and absolutely hold today, whatever he has seen 100 years back. And this is another drawing of Del Rio Ortega. If you see, I personally consider more than science, it's a piece of artwork actually. He has drawn so many cells, and this is the original plate with printed drawings of defined glial cell, but the fecal ligands are not preserved. This fecal ligands has lost in the process. But unfortunately, what happened, 1936, when he was working in a institute, Spain, that time the civil war has been started at Spain. And due to this civil war, he left Spain and moved to England in Oxford University, working in a hospital with a very famous neurosurgeon, Hugh Kearns. So he started working there. As soon as he started working there, his luck was not very good. At least that point of time, his luck is not in favor. The Second World War has been started. And the hospital he was working, German bombed that entire hospital. So he lost probably everything in that point. And I am sure today we have this fecal ligand, probably we could know there are more glial cell types that we could not even discover in last 100 years. And today, if we have the fecal ligands, what Del Rio Ortega has done, maybe there are few more glial cell types which we could know today. But unfortunately, everything lost. And after that bombing happened, German bombing at the Oxford Hospital, Del Rio Ortega left for Argentina. Argentina, after a few years of struggle, he made his own lab around 1936 and 1942, 41. But 1945, he passed away after 3 years. So in Buenos Aires, there is a statue of Del Rio Ortega. If some of you can, I don't know, seen that, but he ultimately passed away in 1945 at Buenos Aires. That's the story of Del Rio Ortega, who discovered the microglial cell 100 years back. But what happened the post Rio Ortega development? The tremendous insight of Rio Ortega, and it is true that it's a tremendous insight. Whatever he has seen in 100 years back, today we verified it is absolutely true even today. With his systematic approach and careful historical descriptions and drawings led him to a series of functional hypotheses which, in spite of the limited techniques available at this time, remain largely true even today. I am sure all of you appreciate the fact, yes, it is remained largely true even today. Surprisingly, however, the field of microglia did not advance for decades, and some neuropathologists even denied their existence for most part of the 20th century. Only 1960s, this German neurophysiologist George Kurzbach group started to study this cell type again with a model called facial nerve axotomy. And after they have started, it's basically a rebirth of microglia research. It slowly picked up. And at present, they are becoming more popular among scientists compared to other glial cells like astrocytes or oligodendrocytes. If you see this data, this is a little bit old, 5 years old. So the people working with astrocytes almost from 1965, 1970, that point. And oligodendrocytes people are start working from 1970, 1975. But microglia as a field actually took a little bit later. It started later, I think around 1987, 88. But in 2015, microglia as a research group across the world crossed the number of astrocytes and oligodendrocytes. So no doubt, it's one of the most popular glial cell and the most popular glial cell, because if you see the worldwide number of PIs or investigation working with glial cells alone, probably there are a maximum number of people working in the microglia. One of the reasons I personally believe, when you work with the oligodendrocytes and the astrocytes, you are all the traditional and trained neuroscientists. But what happened, people working with the microglia, they approach from two different fields, from the neuroscience as well as from the immunology background, because this cell has so much immunological property. Probably that's one of the reasons the number of groups are more compared to other glial cells. So as you can see that these cells actually develop from the periphery. It's not the cell of the brain. So during your development, this cell migrate to central nervous system and monocyte lineage cell and move to CNS during early embryonic development. And during my postdoctoral work, it's a very old paper, and I generally don't cite my postdoctoral work paper, but this paper is very interesting. We showed that neural stem cell in subventricular zone are source of astrocytes and oligodendrocytes, but not microglia. And how this field evolved, if you see, this is the Del Rio Ortega cell in 1999. And then people start doing microglial culture, primary culture, facial nerve axotomy model. And today, with multiphoton imaging system, 2-photon microscope, you can actually image microglia in live animal. And there are so many models people developed in this process, like defined cell lines people are using, BV2, N9, all these popular cell lines. They treat them with microglia with IL-1, beta cytokine, LPS. And people, according to their resource available, they develop so many models. And lots of markers developed in this process, like IBA1 is a very popular marker. And recently, there is another marker available which people think more authentic, TMEM119, which currently we start using our group because IBA1 has some limitations we figured out. That's why we started using this TMEM1199. But this field has tremendously evolved from this Del Rio Ortega cell to today, you can do the live cell imaging by 2-photon microscope. So this is the primary culture of microglia. It's a little bit tricky and time consuming. Generally, graduate students are not very keen to do, from fast go, but ultimately they have to do some point, they have to learn this process. And the way it goes, like 12, 13 days culture. And finally, you get the primary microglia, which is the positive staining for IBA1. And it's a very common technique across the world people are using. We also used in several of our work. And as I mentioned, microglial cell lines are very popular across the world. This is one of the common cell line people are using nowadays, though BV2 is the older cell lines, but N9 is the newer cell lines. So this is one of my recent PhD students cultured this cell line in the lab And finally, with confocal microscope, what she have shown that all these cells are IBA positive. That means they are all microglial cells and their healthy morphology looks they are extremely good condition to work with. So what is the microglia's fundamental or primary function? Primary function, of course, is the innate immune response in the brain. When there is an injury, insult, or infection, microglial cell activated, it starts releasing different cytokine, chemokine, growth factor, increase the vascular permeability, produce nitric oxide, reactive oxygen species, and as a result of that, get the neuronal death. But what happened? This is a very older site I got. I think it is from one of the Helmut Kittelmann's group's review, what I got from maybe 20 years back, almost more than 20 years back, older review actually. So what they have shown here, that depending on the neuronal signal, microglia can respond in two ways. One signal is neuron is damaged but will recover. In that case, microglia can release product like BDNF or CNTF. But there is a second signal which tells that neuron is damaged and beyond repair. So in that case, microglia can release toxic factor like reactive oxygen species or TNF alpha to kill that neuron before damage can spread. So how these two different signal microglia identify? Nobody knows. It's $1 million question. That depending upon the signal, neuronal condition, microglia can either release a factor like BDNF, CNTF, or they can release TNF alpha or reactive oxygen species. And now, most importantly, in last 10 years, there's a tremendous progress in the field. Besides its original role of innate immune response or phagocytosis, now microglia can regulate several normal brain functions. That's why this cell is so amazing or fascinating, because this cell is not in the central nervous system from the beginning, like astrocytes or oligodendrocytes. It's actually in the peripheral cell, migrate to brain in the different parts of development. But how they modulate or how they regulate the brain function, you can see neurogenesis, neural connectivity, synapse monitoring and tuning, myelination, remyelination, vasculogenesis, blood-brain permeability, everything. So that's why this cell is so important, that it regulates almost a lot of function in the central nervous system in disease condition as well as healthy condition. These functions are very close to development. Even 5 years, 6 years back, nobody realized that microglia can play significant role in brain development itself. Now we know it's play a role in brain development by synaptic tuning and synaptic monitoring. Now I will share some of the data from our lab. So redefining innate immunity in the central nervous system and microglia and beyond. Why I put these captions? Because microglia, we know it's a cell in the brain which has the innate immune component. But what I will show, there are some unusual innate immune response in the brain, and that's a different, very unique. Microglia, it is expected they will show certain amount of innate immune response, but there are other cells in brain, even a difficult situation like SOS condition, they can show the innate immune response. So as I mentioned that my group is primarily working with this virus called Japanese encephalitis virus. I'm not going to details of this virus, but what exactly happened following infection, there's a neuronal death because it's a neurotropic virus. And as a result of that, microglia get activated. This activated microglia start releasing cytokine, chemokine, and it's responsible for the neuronal death again. That's we call the bystander killing. And these phagocytic microglia are responsible for cleaning the debris, so they phagocytic the dead neuron. And if you infect the animal, you can see the progressive neurodegenerations in the brain. So this is a Nguyen staining, which shows that following infection, post-infection, there is a less number of neurons are there. And due to this neuronal death, microglia get activated. This is the resting microglia, activated microglia, and this is the phagocytic microglia. And one of my master students, almost 12, 13 years back, showed that not only these cells are activated, they also release different cytokine, chemokine following the infections. Now come back to the very recent time. I mean, I'm showing mostly last 4 or 5 years of data. A postdoc in my lab showed that this microRNA also upregulated following infection in these microglial cells, like defined microRNA. And I will talk about the one microRNA here, microRNA 301A. Another postdoc in my lab showed that this microRNA 301A upregulated in microglial cells like BB2 in late time point and neuron in the early time point. And based upon his work, what he has shown that this microRNA 301A regulates inflammatory response to JE virus following biosuppression of this NK-RF activity. I'm not going to details of this work, but why I'm showing this work, and this work also done with the collaboration with NIMHANS. We did some extensive staining in the autopsy brain tissue sample, which my good friend and colleague from NIMHANS, Dr. Anita Mahadevan, provided us. So in this paper, one of the important findings we have that JV infection induces classical activation of microglia that drive the production of pro-inflammatory cytokine while suppressing alternative activation that could serve to dampen the inflammatory response. So why it is so important? Because immunologists knows for a long time that there are two types of monocyte or macrophages, M1 and M2. M1 is more pro-inflammatory, M2 is less pro-inflammatory or anti-inflammatory. But now microglia, people have these suspicions for a long time, maybe microglia also have the two different types. And in this paper, we showed very clearly, yes, microglia also have the two defined phenotypes like M1 and M2. And we proved that with different markers of M1, M2. Now, advantage is that if microglia is the responsible for inflammation, so you can target the particular microglial cell. But the problem is that, as you know now, that microglia besides inflammation do other normal brain function. So it's not really right to target all the microglial cell in the population. So it is important that now we know to define microglial phenotype M1, M2. So you can target the M1 phenotype, not the M2. M2 actually giving the protection to the host because it's anti-inflammatory phenotype. But M1 is more pro-inflammatory. So you have to target any microglial population, you target the M1 population. So I'll just shift gear a little bit. Our lab, one of the things we are also interested that how virus infect the neuronal stem progenitor cell, because we have shown that this virus block the neural stem cell proliferation. Not only that, it actually modulates the differentiation pattern of this neural stem cell progenitor cell. So my student Shulagna, almost 12 years back, 11 years back, showed that this virus indeed infect neural stem cells in the brain, and neural stem cells is a neural progenitor cell which stay in the subventricular zone. And the marker for neural stem cell is a nestin. And we have shown here that there are cells colocalized for both the virus and for the nestin. That means this particular cell is the neural progenitor cell which is infected by the virus. And when she studied in the animal model, here is the surprise. We have seen that all the molecules like CD40, CD80, CD86, MSC class 1, they are basically antigen presenting cells molecules. These molecules are normally present in dendritic cells, macrophage, monocyte, or in B cells. But what interestingly happened that following infection, neural stem cells, because all these are marker positive for the nestin, as you can see here, which is a marker for stem cell. This CD40, CD80, CD86, MSC class 1, they are expressed in this cell. That means it's the unusual innate immune response. Now, following infection, neural stem cell shows the marker for antigen presenting cell, professional antigen presenting cell like dendritic cells or monocyte. Moreover, another important tool we used in our lab, we cultured neuronal progenitor cell from the brain. And what we did here, we developed, following culture, we have this neurosphere, and neurosphere is all neural stem cell, which is positive for nestin and Musashi. And for this neural progenitor cell, you can use the appropriate growth condition in the media, you can have either neurons or astrocytes or even oligodendrocytes, depend upon that, what culture or growth condition you are using. So Shulagna, what she did the ex vivo experiment. She cultured these neuronal stem progenitor cells and treat them with virus, then see the expression of this CD40, CD80, CD86, MSC class 1. And here also she has observed that this neuronal stem cell express all these markers for antigen presenting cell like CD40, CD80, CD86, and MSC class 1. And when she did flow cytometry with this neuronal progenitor cell, they are also same thing she observed, that they express 1 day, 2 day, 3 day, 4 day post-infection like CD40, CD80, CD86, MSC class 1. But these markers are not supposed to present in the neuronal stem cells. As I mentioned, this is a typical marker for antigen presenting cells or the immune competent cells. Moreover, not only they express these antigen presenting molecules, they are also functionally active. When she cultured them and see the cytokine level, all these neuronal progenitor cell produce TNF alpha, interferon gamma, IL-6, and CCL2, the chemokine. So they also make this cytokine following the infection. Lastly, this is a very popular technique people are immunologists are using for a long time. It's called allogenic mixed leukocyte reaction. So you take antigen presenting cells from one genera of mice like a black mice and take T cell from white mice like Balpsy mice and culture them, then T cell proliferate. So that's the called allogenic mixed leukocyte reaction. What she did here, she treat the neuronal stem progenitor cells, which is derived from a Balpsy mouse, and treat them with the virus, and then take T cell from C57 black 6 mice and culture them. Here you can see in the control group, the T cell proliferation is a 9.7%. And when he did that with the J-infected neuronal stem progenitor cell, it is 18.1. That means the T cell proliferation doubled. So that shows that neuronal stem progenitor cell actually acting as a professional antigen presenting cell. That's very unlikely. The typically behavior of a dendritic cells or monocyte. But now in a situation, neural stem cell progenitor cell also kind of behave as antigen presenting cells. Lastly, what she showed that if you take these neural stem progenitor cells, they also make interleukin-2. And why interleukin-2 is important? Interleukin-2 is a growth factor for T cells. Now, in a conclusion, what she said that this neuronal stem cell progenitor cell can really define cytokine, chemokine, and bring other cells from the peripheral system by breaching blood-brain barrier. Not only that, this stem cell progenitor cell can make interleukin-2, which is a growth factor for T cells. So there are more T cells are growing in the system. So that means in a situation when immune system is compromised, then this neural stem cell progenitor can actually act as a immune competent cell. So this is a SOS call. It's normal situation, it may not happen, but in a SOS situation, when infection happens, this stem cell probably could act as a antigen presenting cells. Now, another unusual innate immune response I mentioned, and that is my last part of my talk, that neuron can act as an innate immune component. So one of my PhD students, Arshad Nazmi, showed that following viral infection, some of the cells, and these are the neuronal cells which we identify that they can express STING or RIG. The STING or RIG, both are immune adapter molecules that are important for the immune competent of the cells. Not only that, with the primary cortical cells following infection, he has found that they express RIG1. And we have some data from the autopsy brain tissue sample from NIMHANS that yes, indeed, in patient autopsy brain tissue sample, there is the expression of the TLR7, which is also important for the innate immune component. Lastly, I will go back to the slides which I showed earlier, that this a microRNA expressed in the early time point in the neuronal cell. I have showed the data from the microglial cell, but now in the neuronal cells, this microRNA expressed in the early time point. Another postdoc in my lab, Bhivavasu Hajira, showed that this neuron actually expressed interferon beta. This is a very unusual condition again. neuron not supposed to express this interferon response, but when a situation happens that there is a SOS call, that immune system is compromised, that neuronal cell can act as an interferon beta response. And one of the reasons that this is probably the first evidence that a neuronal cell can actually make the interferon response, the journal decided to put this in the cover page image. We also showed that not only neuron, there are transcription factors which is important for the interferon response, like IFIT1, ISG, and OS1 also expressed in these neuronal cells. I think this is my last slide. This is my acknowledgement. I'm very lucky to have a wonderful group of colleagues and my collaborators. We are working for a long time. I'm not going to take any of their names. They are all wonderful scientists and wonderful people and wonderful human beings. Last but not the least, my students. So this is the current group of students. I think this is the photo from last 1 year. With that, I would like to thank Indian Academy of Neuroscience one more time for considering for this Oration Award.
Thank you, Dr. Anirban Basu, for this wonderful oration. And it was truly, I felt it is a truly an educational entourage in the world of GLIA. And you have very lucidly put it, like the role of GLIA in immune response and also the neurons in immune response, right from the historical perspective to the function, and then focusing on your work. Beautiful, very beautiful entourage, and we really congratulate you for delivering this oration. Yeah. Over to Dr. Anita Jagota for taking up, if possible, because orations we usually don't encourage.
Don't encourage questions.
But still, Anita, you can decide. Take it.
I did the whole this thing to the chairperson, and as per the constitution of IAN, if
usually such oration, they say that you can have some discussion during the tea break time, or so usually this kind of oration is kept just before tea so that they can go together and have a better discussion. So, but anyway, I think that the chairperson is the person who can decide about it.
Yeah.
If one or two question is there and Dr. Basu is ready to take it, I think they can do it.
I am fine to take the question.
Yeah.
Yeah, one question I found from one of the students that during JE virus infection, what are the markers you have used for this identifying this M2 phenotype? Like,
there are lots of markers. Actually, it's not. I mean, I think they are interested if you read that paper. So it's a lots of markers. It's not the one marker actually.
Okay.
So we have lots of markers, because this is, as I mentioned, that this is one of the first paper, I think, for the microglial cell. Monocyte macrophages people showed for a long time, but microglia is the first paper which clearly showed that the presence of two different subtypes in the brain, microglial subtypes. So to prove it, we actually did a very thorough work actually.
Yeah, thank you, Anita. And there is one question from Dr. that have you seen any astrogliosis in this?
Yes. We astrogliosis we have seen. That's a different work. Actually, that's one of my student work also. But astrocytes I didn't show it. Yeah, astrogliosis you can see it.
Yeah, this is from Dr. T.R. Raju. I think, hearty congratulations, Dr. Anirban, for this lucid presentation. And
thank you so much.
We will move on to Dr. Anita now.
Congratulations, Anirban.
Yeah, thank you.
Dr. Anita?
As always, great talk, Anirban.
Thank you.
Congratulations, Anirban.
Thank you.
Congratulations, Dr. Anirban. This is Vinay Khanna.
Thank you. Yeah, I've seen.
Sorry, we are not able to felicitate physically.
Yeah, physically we are not. Next year.
Accept our congratulations, and we will request you to be there during the next IN meeting, where we like to felicitate there itself.
Yeah.
Sure.
We'll inform you the dates later.
Okay.
Thank you.
So this is the tea time then, interactions going on.
Thank you, Professor Anirban Basu sir, and also Professor Bindukuti ma'am for this wonderful session. And we really enjoyed sir's oration lecture and the interaction, which is normally not allowed, but with the kind permission of Professor Bindukuti ma'am and Professor Anirban Basu, we had the interaction as per suggestion of Professor Agrawal sir. And now we will have another session, exciting session again on Young Scientist Colloquium. But before that, I have to make an announcement that let us take 10 minutes break, and on the same link, we will continue at 8:05.